In 1 teaspoon of our blood, there are approximately 5 million killer T cells, which are naturally created to execute a targeted elimination of cancer cells or infected cells with precision, without harming other normal cells at all. When viewed under a microscope with filtered light, where a killer T cells appears green and a cancer cells appears blue, you can see that when the killer T cells injects a substance into the blue cancer cells, the blue cell immediately disintegrates. In the illustrated simulation of its function, a killer T cell is seen attaching to a cancer cell. Upon encountering this cancer cell, it sends out signals to call for reinforcements. It then releases perforins to create pores in the cancer cell and injects granzymes, which are brown substances, into the cancer cell to dissolve its proteins until the cancer cell eventually disintegrates and disappears.